You've found Dreamtime Science, part classroom, part lullaby, entirely devoted to satisfying your curiosity while you find your way to sleep. You know what's fascinating? I've been thinking a lot about how our bodies seem to have their own little conversations happening all the time, especially between our gut and our brain. There's something remarkable about this second brain tucked inside us, quietly sending messages that shape how we feel and think, and I can't wait to share what scientists are discovering about this hidden connection. Should these dreamy explorations accompany your journey to sleep, a gentle like or subscription would be deeply appreciated. What questions about our universe keep you wonderfully curious? Now, take a moment. Adjust your blanket. Find the perfect hollow in your pillow. Let your shoulders melt. Breathe in and out. Beneath the surface of your daily life, quietly, persistently, something stirs, a presence so familiar that you rarely notice it until it shouts, a peculiar flutter, a tightening coil, a gentle warmth or a cold hollow deep in your abdomen. Sometimes it comes as anxiety before a difficult conversation, the thrill before a long-awaited reunion, or a warning in the hush before a storm. Ancient philosophers, poets, and healers have all recognized this odd, wordless messenger, naming it intuition, instinct, or simply a gut feeling. For centuries, humanity has looked to the gut as a place of wisdom, a silent oracle humming with foreboding or calm. In the markets of ancient Athens, traders would clutch their bellies and claim to sense a bargain's dishonesty. In Chinese medicine, the belly, known as the dantian, is a crucible of qi, the life force. In the lore of the Vikings, sagas tell of warriors who trusted their magafylgja, the guiding spirit residing in the stomach, to steer them through peril. And even now, in the language of every culture, the gut persists. I have butterflies in my stomach. Now go with your gut. It was a gut-wrenching decision. These phrases echo through time, carrying with them the conviction that something hidden within us knows more than we do. What is it about this soft, dark, coiling mass of organs that makes us believe it can whisper truths about the world? Why, of all places, would the seat of our deepest knowing reside in the folds of our intestines, among the silent churn of digestion? The heart, after all, beats with the rhythm of life and love. The head sparkles with reason and thought. Yet it is the gut that seems to pulse with instinct, to answer questions we have not yet asked. For so long, this intuition remained a mystery, an oddity, perhaps, or a trick of the mind. The great thinkers of the Enlightenment, with their love of reason and clarity, dismissed gut feelings as primitive echoes, the leftovers of an animal past. The gut, they insisted, was a mere machine, a tube for food, a chamber for waste, an engine for survival. Its gurgles and growls were only the noises of biology, nothing more. But the myths endured. They endured because, time and again, the gut proved itself to be right. A mother senses danger before seeing the threat. A chess player, after long hours of study, knows the perfect move without calculation. An explorer, on the edge of the unknown, turns back at the last moment, spared from disaster by an uneasy twist in their belly. These are not just stories. They are experiences, woven into the fabric of human life, impossible to dismiss. As the modern world unfolded, the myth of the gut took on new forms. In the 19th century, as scientists peered into the body's inner workings, they found that the intestines were lined with a vast network of nerves. The enteric nervous system, they called it, a dense web of neurons, hundreds of millions strong, twining through the digestive tract from esophagus to colon. Even then, some suspected it was more than a simple relay. But for over a century, this second brain remained in the shadow of the first, overshadowed by the glittering cortex, the pride of human evolution. Yet the question lingered, persistent and soft as breath. How could mere nerves in the gut give rise to feeling, to intuition, to a sense of knowing? As scientists pressed deeper, they began to unearth a secret world beneath logic and thought, a world where the gut is not only a machine, but a messenger, a whisperer, a subtle guide. Close your eyes for a moment, and let your attention drift down from your skull, through your throat, past the steady pulse of the heart, into the warm, shifting landscape of your abdomen. There, beneath skin and muscle, the gut coils upon itself in silent labor. Each day it turns and transforms the food you eat, extracting energy, building blocks, and discarding the rest. But behind this mechanical dance, something else is happening, an endless conversation carried out in pulses of electricity and tides of chemicals between gut and brain. This conversation is not one-sided. The brain, of course, commands the gut: quicken, slow, contract, release. But the gut, astonishingly, speaks back. It sends signals of its own, encoded in neurotransmitters like serotonin and dopamine, the very same chemicals that color our moods and dreams. In fact, most of the body's serotonin is not made in the skull, but in the lining of the gut, bathed in the swirl of digestion, touching every cell of the enteric nervous system. Here, the gut is not just a receiver of orders, but a source of sensation and feeling, a generator of moods. The second brain theory, as it has come to be called, is not a theory in the sense of idle speculation. It is the slow, careful unveiling of a reality long sensed but only recently understood. The enteric nervous system contains over 500 million neurons, more than the spinal cord. It can operate independently, orchestrating the complex ballet of digestion even if its connection to the brain is severed. It reacts to stress, to pleasure, to the presence of food or the threat of poison. It remembers, in its own way, the patterns of nourishment and danger, and it whispers these memories upward in the language of sensation, emotion, and yes, intuition. To understand this second brain is to step into a realm where the boundaries between mind and body begin to blur. Here, the gut is not merely a servant of appetite, but a participant in the story of who you are. When you feel a knot in your stomach before public speaking, or a wave of relief after hearing good news, you are experiencing the echoes of this conversation. The gut listens to the world, translates it into chemical messages, and shares its knowledge with the mind. This is not to say that the gut is clairvoyant, or that intuition is infallible. The signals it sends are shaped by evolution, by experience, by the millions of microbes that call your intestines home. Sometimes the gut errs, misreading signals, conjuring anxiety where none is needed. Yet, often enough, it is uncannily right. The body, after all, is a creature of patterns, forever learning, adapting, remembering. It knows the taste of safe food, the rhythms of trust and threat, the subtle cues that escape the conscious mind. In the 20th century, as neuroscience blossomed, researchers like Michael Gershon and Jackie Wood began to map the intricacies of the enteric nervous system. They discovered that the gut's neurons form circuits, loops of activity that can sustain themselves, process information, and coordinate responses. These circuits are bathed in a sea of neurotransmitters, sensitive to the faintest changes in chemistry. When you eat, the gut senses the composition of your food and decides how to digest it. When you are afraid, the gut tightens, preparing for flight or fight. Even in the absence of the brain's direct control, the gut can sense, decide, and act. This autonomy is not absolute, but it is profound. The vagus nerve, a thick bundle of fibers winding from brainstem to colon, acts as a highway for information, conveying messages in both directions. Through the vagus, the gut tells the brain about fullness, pain, discomfort, or delight. Some 90% of the nerve fibers in the vagus carry information from gut to brain, not the other way around. The implications are staggering. The mind, it seems, is not an isolated captain steering the ship of the body, but a listener, forever attuned to the stories told by the gut. And what of those stories? They are ancient. Before language, before culture, even before the emergence of a true brain, creatures sensed the world through their guts. The earliest animals, simple, soft-bodied things, felt the presence of food, the touch of poison, the warmth of sun, or the chill of danger, not in a head, but in the tissues of their bodies. Over millions of years, these sensations were woven into networks of nerves, patterns of response, the seeds of what would become emotion and thought. In this light, our modern gut feelings are echoes of a deep evolutionary past. They are the wisdom of countless generations, distilled into a shiver of unease or a bloom of contentment. The gut is not infallible, but it is ancient, shaped by survival, honed by experience. It watches the world, senses its rhythms, and tries to keep you safe, even when you do not know why. The myths of the gut, then, are not merely superstitions. They are attempts to name and honor this silent wisdom, to regard the body not as a machine, but as a living, sensing being. When the Greeks spoke of the phrenes, the seat of thought and feeling in the belly, they were, in a sense, acknowledging the truth that science would only later unveil. The gut is a place of knowing, a center of feeling, a second brain. But how does this second brain shape our lives? What does it mean to live in a body where feeling and thought are entwined, where messages from the gut can sway decisions, color moods, or guide us through danger? And what of the strange, invisible residents of the gut, the trillions of microbes that dwell within us, shaping our health, our cravings, even, perhaps, our minds? As the 21st century unfolds, the science of the gut is entering a golden age. New tools allow us to peer into the tangled networks of the enteric nervous system, to trace the flow of neurotransmitters, to listen in on the silent conversations between gut and brain. Researchers are beginning to understand how the gut senses the world, how it remembers, how it learns. They are mapping the ways in which the gut's whispers shape not only our health, but our happiness, our fear, our choices. Yet for all this progress, the gut remains a place of mystery. Its signals are subtle, its messages often ambiguous. It speaks in the language of sensation, not words, in feelings, not logic. To listen to the gut is to cultivate a kind of attention that is both ancient and new, a willingness to trust the body's wisdom, even when it cannot be explained. So tonight, as you lie in the quiet dark, perhaps you will notice the gentle stirrings in your own belly, the slow rise and fall, the secret dance of nerves and chemicals, the quiet conversation unfolding just out of reach. These are the whispers of your second brain, the ancient messenger, the guardian of your hidden knowing. If you listen closely, you might begin to sense its presence, not as an oracle, not as a machine, but as a companion on the journey of your life. For in the winding corridors of the gut, in the secret language of nerves and cells, a story is being told, a story that is yours and older than you, and still so very much unfinished. And as we drift further into the labyrinth of the second brain, we find ourselves at the threshold of another mystery, where not only nerves and chemicals, but entire worlds of invisible life shape the whispers within. The story, it seems, is only just beginning to unfold. Beneath the surface of our daily awareness, a ceaseless dialogue unfolds, a conversation older than our species, conducted in the secret language of molecules and electricity. The gut and the brain, two organs separated by the length of the torso, are partners in a complex, dynamic ballet. This relationship is neither simple nor completely mapped. It is an intricate web of signals, a dance of mutual influence and sometimes profound mystery. To understand its complexity is to step into a world where the boundaries of the self are blurred, where bacteria whisper to neurons, and where emotions can ripple through the lining of the intestine like wind over grass. Imagine, for a moment, the gut not as a mere digestive vessel, but as a bustling metropolis, its walls lined with trillions of inhabitants, bacteria, viruses, fungi, each one a participant in the metabolic and chemical symphony of life. This collective, often called the gut microbiome, is as diverse as a rainforest and as dynamic as a city square at dawn. Each microbe contributes something unique. Some ferment fibers into short-chain fatty acids, others synthesize vitamins or break down toxins. Yet their influence does not end at digestion. Instead, their metabolic byproducts seep into the bloodstream, drifting upward toward the brain, messengers in the body's internal mail. This is the beginning of the unseen conversation. Chemical signals generated by microbes can modify the function of the nervous system, and in return, the brain can shape the gut's environment through its own mechanisms, stress hormones, for example, or the modulation of gut motility. The link between these distant organs is often referred to as the gut-brain axis, a term that conjures images of a single cable, perhaps a telephone wire, quietly humming with information, but the reality is far more elaborate. At the center of this dialogue lies the vagus nerve, a slender, wandering cord of nerves that stretches from the brainstem, weaving through the chest to the intricate folds of the gut. It is a bidirectional superhighway, carrying not only sensory data from the gut upward, but also motor instructions and regulatory signals from the brain downward. When you feel butterflies in your stomach before speaking in public, or the hollow ache of anxiety after a sleepless night, the vagus nerve is the courier, translating emotion into visceral sensation. Yet the vagus nerve is only one channel among many. There are endocrine signals, hormones released by gut cells in response to the arrival of food or to changes in microbial composition. These hormones, such as ghrelin, which stimulates appetite, or peptide YY, which signals fullness, can cross the blood-brain barrier, influencing mood, hunger, and even cognition. There are also immune signals, as the gut is both a barrier and a gateway, sampling the world outside through food and drink, and alerting the brain to the presence of pathogens, or the soothing presence of friendly microbes. But for all the channels we have identified, and all the patterns we have begun to discern, the true complexity of the conversation remains elusive. Every year, new discoveries reveal fresh layers of intricacy. This is not a tidy dialogue, it is more like a vast polyphonic chorus, with countless voices rising and falling in unpredictable harmony or discord. Take, for example, the role of serotonin, a neurotransmitter commonly associated with mood. Roughly 90% of the body's serotonin is produced not in the brain, as one might assume, but in the cells lining the gut. This serotonin does not cross the blood-brain barrier directly, yet it can influence the central nervous system through indirect pathways, affecting everything from gut motility to emotional well-being. The bacteria in the gut can affect how much serotonin is produced, and in turn, the levels of serotonin can influence which bacteria thrive. The relationship is not one-directional, but an ever-evolving feedback loop. The immune system, too, is deeply entwined with the gut-brain conversation. The gut is the largest immune organ in the body, constantly sampling antigens from food and microbes. When this system is in balance, it protects us from infection and inflammation. But when disrupted, through infection, stress or diet, it can send distress signals to the brain, triggering changes in mood and behavior. Inflammation in the gut has been linked to depression, anxiety, and even neurodegenerative diseases, yet the precise mechanisms remain shrouded in uncertainty. Are these changes driven by immune molecules called cytokines, by microbial metabolites, or by as yet unknown factors? This uncertainty is a recurring theme. The more we learn, the less certain we become of where the boundaries lie. For decades, it was assumed that the brain was the master controller, dictating orders to the rest of the body. But the gut, with its own nervous system, the enteric nervous system, sometimes called the second brain, challenges this hierarchy. The enteric nervous system contains as many neurons as the spinal cord and is capable of autonomous function. It can coordinate the rhythmic contractions of peristalsis, manage secretions, and even generate feelings of discomfort or well-being, all without direct input from the brain. This independence raises profound questions. When you feel a gut instinct, is it merely the brain interpreting signals from the digestive tract, or is the gut itself generating a form of intelligence? Some researchers speculate that the gut's neural networks evolved to manage the complex demands of digestion long before the brain took on its modern form. Others suggest that the gut's autonomy is a relic, a vestige of our evolutionary past, when survival hinged as much on efficient nutrient extraction as on sensory perception. What is clear is that the gut and brain are engaged in a dynamic, reciprocal relationship. Changes in the gut, whether through diet, infection, or microbial imbalance, can alter mood, cognition, and even personality. Conversely, stress, trauma, and emotional upheaval can reshape the gut's landscape, changing which microbes flourish and which fade away. This circular causality complicates the search for simple answers. Is depression a cause or a consequence of gut inflammation? Does anxiety originate in the brain, or is it rooted in microbial signals from the intestine? The answer, more often than not, is both. Despite a growing body of research, much remains uncharted. Many studies rely on animal models, mice with simplified microbiomes, or germ-free rodents raised in sterile environments. These models reveal tantalizing clues. Mice with disrupted gut flora show changes in anxiety-like behavior, in memory, even in social interaction. But translating these findings to humans is fraught with complexity. The human microbiome is vastly more diverse, shaped by diet, environment, genetics, and chance. Two people living in the same household may share only a fraction of their gut microbes, and the functional consequences of these differences are only beginning to be understood. Moreover, the tools we use to measure the microbiome, sequencing technologies, metabolomic assays, are powerful but limited. They tell us which microbes are present and sometimes what they are doing, but they cannot yet capture the full richness of microbial communication. Many gut bacteria cannot be cultured in the lab. Their metabolic pathways remain obscure, and the interplay between microbes, host cells, and the nervous system is a labyrinth of feedback and modulation. It is as if we are eavesdropping on a conversation in a language we only partly understand, catching fragments of meaning but missing the nuances and the subtext. Amidst this complexity, the allure of simple solutions remains strong. Probiotics, prebiotics, dietary interventions, each promises to reshape the gut-brain dialogue in beneficial ways. And indeed, there is evidence that certain strains of bacteria can improve mood, reduce anxiety, or enhance cognitive function. But the effects are often modest, inconsistent, and deeply individual. What works for one person may fail for another, and the reasons why are hidden in the tangled web of genetics, lifestyle, and microbial ecology. Complicating matters further are the many variables that can affect the gut-brain axis. Antibiotics can wipe out entire populations of gut bacteria, sometimes with lasting consequences. Stress can alter the permeability of the gut lining, allowing molecules to pass into the bloodstream that would otherwise be excluded. Diet can feed beneficial microbes or starve them, reshaping the ecosystem with each meal. Even the time of day, the rhythm of sleep and wakefulness, can modulate the signals exchanged across the axis. And then there are the outliers, the cases that defy easy explanation. There are people who, after a bout of food poisoning, develop chronic changes in mood or cognition, phenomena sometimes described as post-infectious syndromes. There are those whose depression lifts following a change in diet, or whose anxiety abates after a course of probiotics. These anecdotes, while not definitive, hint at connections that are only beginning to be mapped. The limitations of our understanding are not simply technical, they are conceptual. The reductionist approaches of classical science, isolating variables, seeking linear causality, are ill-suited to the tangled complexity of the gut-brain axis. Here, causes and effects loop back on themselves, and the whole is far more than the sum of its parts. To truly comprehend this relationship, we may need new models, networks, systems, ecologies, capable of capturing the multiplicity of pathways and the emergent properties that arise from their intersection. Yet within these limitations lies a kind of beauty. The very complexity that frustrates our attempts at control is also a source of resilience and adaptability. The gut-brain axis is not a brittle mechanism, but a living system capable of absorbing shocks, adjusting to change, and sometimes healing itself. It is a reminder that we are not isolated minds piloting inert bodies, but participants in a web of life that extends from the soil beneath our feet to the neurons firing in our skulls. This perspective invites humility. We are only beginning to listen to the conversation within, to decode its myriad voices and understand their meaning. Each answer begets new questions. How do early life experiences, such as birth by caesarean section or breastfeeding, shape the gut-brain axis for a lifetime? What role do viruses play, those invisible hitchhikers that may outnumber bacteria by 10 to 1? How does the gut's conversation with the brain change with age, with illness, with the tides of our internal and external worlds? As you settle deeper into the softness of the night, the mystery of the gut-brain axis becomes a gentle background hum, a reminder that within every body a conversation is unfolding, ancient and ongoing, more intricate than any story we might tell. The surface may seem calm, but beneath, the currents swirl in patterns not yet fully revealed. And so, the unseen conversation continues. Tomorrow's discoveries may cast new light on old questions, or reveal unsuspected connections. For now, we are left with the wonder of complexity, the allure of what lies just beyond the edge of understanding. In this space between knowing and unknowing, the story lingers, poised for the next chapter, where the signals of the gut and the thoughts of the mind entwine ever more deeply, awaiting the dawn and the promise of further revelation. Throughout the corridors of scientific inquiry, there are few conversations as intricate and elusive as the one whispered between the gut and the brain. This dialogue, woven from threads of chemistry, electricity, and living cells, unfolds largely beyond the reach of our conscious perception. Yet, behind the closed doors of laboratories, in the hush of midnight-lit research centers, and within the hum of analytic machines, scientists have devoted themselves to unraveling the language of this internal exchange. To eavesdrop on the gut-brain connection is to become a detective of the body's most delicate secrets, armed with an ever-evolving arsenal of tools and techniques designed to peer into the depths of our own biology. To begin, let us drift into the world of imaging, a realm that allows us to see what was once invisible. One of the earliest and most transformative tools in this domain is functional magnetic resonance imaging, or fMRI. With the patient lying quiet and still within a giant humming magnet, scientists can capture snapshots of the brain in action, seeing which regions light up in response to a meal, a thought, or the flutter of a signal from the gut. fMRI does not see neurons directly, but instead measures changes in blood flow, a proxy for neural activity. When the gut sends its silent signals, perhaps in the aftermath of a nourishing meal, or the uneasy churn of anxiety, the brain's response is painted in subtle strokes across the monitor, a bloom of activity in the insula, the amygdala, the prefrontal cortex. Patterns emerge, sometimes surprising, hinting at the routes along which the gut and brain converse. But watching the brain alone is only half of the story. Scientists yearn to map the dialogue itself, tracing the signals as they travel from the twisting coils of the intestines up the bundled wires of the vagus nerve and into the sanctuary of the skull. To do so, some turn to electrophysiology, the science of recording the electrical language of nerves. Tiny electrodes, some as fine as a human hair, can be placed along the vagus nerve in experimental animals, capturing the rapid-fire bursts of electricity as information flows. In these studies, a mouse might be fed a rich, fatty morsel or given a drop of bitter liquid, while the electrodes whisper back to the researchers, here a signal of satiety, there a pulse of discomfort. The timing, the frequency, the amplitude all become clues in the deciphering of this ancient code. Yet the gut is not merely a passive sender of messages. It is an ecosystem unto itself, home to trillions of microbes whose chemical chatter influences both the gut lining and, indirectly, the mind. To probe this microbial world, scientists employ the meticulous practices of microbiome analysis. Fecal samples, carefully collected and preserved, are subjected to DNA sequencing, revealing the identities and relative abundances of the bacteria, viruses, and fungi that dwell within. With further layers of analysis, metabolomics, for instance, researchers can track the chemical byproducts of microbial metabolism, short-chain fatty acids, neurotransmitter mimics, fragments of cell walls. These molecules, born from the digestion of fiber or the breakdown of proteins, can act as messengers, crossing the gut barrier and influencing distant organs, including the brain. Within these samples lie the fingerprints of health and disease. In some experiments, scientists compare the microbiomes of individuals with and without depression or anxiety or neurodegenerative diseases, searching for patterns. A missing genus here, an overabundance there. To test causality, the field employs a remarkable technique, fecal microbiota transplantation. By transferring the gut microbes from one animal to another, sometimes even from humans to mice, researchers can observe changes in behavior, metabolism, or brain chemistry. A mouse that receives the microbiota from a depressed human, for instance, may become less exploratory, more anxious, as if inheriting not just bacteria, but a fragment of emotional state. In these studies, the gut and mind are revealed as conjoined twins, each shaping the other through the medium of microbes. To further untangle the web of causation, scientists reach for the precision of genetic and molecular tools. In the realm of optogenetics, for example, specific neurons within the gut's own nervous system, the enteric nervous system, sometimes called the second brain, can be made sensitive to light. By shining a pinpoint of laser through a fiber optic cable, a researcher can activate or silence individual neurons in a living animal, observing how the gut's movements, the animal's feelings of hunger, or even its anxiety levels, are altered. This is an act of exquisite control, a way to test hypotheses about which neural circuits are responsible for which sensations. Chemogenetics provides another avenue using designer drugs to activate or inhibit genetically modified receptors. With careful planning, an experimenter can selectively quiet the chatter of certain gut neurons or ramp up their activity, all from the safety of a laboratory bench. The effects ripple outward. Sometimes the animal eats more, sometimes less. Sometimes stress responses are blunted, sometimes heightened. Each experiment is a brushstroke on the vast canvas of the gut-brain dialogue, filling in details that were once only guessed at. Yet the gut's conversation with the brain is more than just nerves and microbes. It is a chemical symphony conducted across barriers and through the bloodstream. To track these molecular messengers, scientists employ the delicate arts of mass spectrometry and chromatography. Blood samples, drawn at intervals after a meal or a stressor, are spun in high-speed centrifuges, separating plasma from cells. The plasma is then introduced into machines that can separate, identify, and quantify thousands of molecules at once: peptides, hormones, metabolites. Among these are old friends: serotonin, much of which is produced in the gut; ghrelin, the hunger hormone; leptin, a signal of fullness; and a host of lesser-known players whose roles are only beginning to be understood. By mapping how levels of these molecules rise and fall, how their presence correlates with mood or cognition, researchers gain insight into the invisible threads that tie our bellies to our brains. Some experiments are more direct still. In clinical research, volunteers might ingest a capsule containing a temperature or pH sensor, which wirelessly transmits data as it journeys through the digestive tract. Others may swallow a tiny camera, a pill cam, that snaps images of the gut's inner landscape, revealing the rhythmic contractions of peristalsis or the subtle inflammation of mucosal linings. Coupled with questionnaires, cognitive tests, and even real-time monitoring of heart rate or skin conductance, these studies allow scientists to correlate gut events with emotional or cognitive states. Do certain foods provoke anxiety in sensitive individuals? Does the gut's response to stress leave a lingering fingerprint in the mind? The data, once collated and analyzed, whisper hints of answers. In an even more controlled fashion, animal studies offer the power to manipulate individual variables with almost surgical precision. Germ-free mice, raised in sterile conditions without any gut microbes at all, provide a blank slate. When these mice are colonized with specific strains of bacteria or mixtures drawn from human donors, their development can be tracked in detail, from the wiring of their brains to the subtleties of their immune responses. Some germ-free mice display altered anxiety behaviors, impaired cognition, or even changes in pain sensitivity. Through such experiments, the necessity and sufficiency of the microbiome in shaping the gut-brain dialogue becomes starkly clear. Of course, not all experiments are so invasive. Some techniques rely on the careful observation of natural variation. Population studies, drawing on the medical records and biological samples of thousands, allow researchers to tease apart correlations between gut health, diet, mental states, and disease. Longitudinal studies, following individuals over years or even decades, can reveal how early life events, antibiotic use, or dietary shifts alter the gut-brain axis, sometimes with effects that echo across a lifetime. In these vast data sets, clever statistical tools and machine learning algorithms are deployed, searching for patterns too subtle for the human eye to see unaided. A powerful complement to these empirical approaches is the construction of in vitro models, miniature simulations of the gut-brain dialogue outside the body. In the laboratory, cells from the gut lining can be grown on permeable membranes, forming a barrier that is then exposed to microbial metabolites or immune signals. On the other side, neurons derived from stem cells may be cultured, their electrical activity monitored as new molecules are introduced. These organ-on-a-chip systems, while simplified, allow for the dissection of mechanistic pathways. Which molecules cross the gut barrier? How do they alter neuronal firing? What happens when inflammation arises or when the microbial community shifts? With each experiment, a piece of the puzzle snaps into place. Yet, perhaps the most remarkable tool of all is the human itself, the subjective experience of sensation, emotion, and thought. In clinical studies, patients may be asked to rate their feelings of fullness, anxiety, or discomfort, sometimes in response to specific interventions. These subjective reports, when paired with physiological measurements, close the loop, allowing for the mapping of internal states to the objective data gathered by machines and assays. In some cases, biofeedback techniques are used, training individuals to modulate their own gut activity or stress responses with conscious effort. The boundaries between body and mind are blurred, and the gut-brain dialogue becomes not just a matter for the laboratory, but a lived experience. As the tools have grown more sophisticated, so too has the ambition of the experiments. Multi-omics approaches, integrating data from genomics, transcriptomics, proteomics, and metabolomics, allow for the holistic mapping of the gut-brain axis, capturing its complexity in all dimensions. Advanced imaging, such as positron emission tomography, PET, can trace the movement of labeled neurotransmitters or hormones through the body in real time. New generations of wireless sensors promise to deliver continuous monitoring of gut activity, opening the door to studies that unfold not just in the clinic, but in the everyday lives of participants. There are, of course, limitations and challenges. The gut's environment is dynamic, ever-changing with each meal, each stressor, each microbial bloom. The brain, too, is subject to the vagaries of mood, sleep, and experience. Correlation does not always mean causation, and the signals are often faint, buried beneath layers of noise. But with each new tool, each clever experiment, the picture becomes a little clearer. The dialogue between gut and brain, once thought to be a mere background hum, is revealed as a symphony, complex, adaptive, and deeply intertwined with the story of what it means to be alive. It is a story that is still being written, its pages spread across the journals of neuroscience, gastroenterology, microbiology, and beyond. The questions multiply. Can we harness the gut-brain axis to treat mental illness, to promote healthy aging, to understand the origins of consciousness itself? The tools are in place, the experiments ongoing, the possibilities vast. And yet, as one stands at the threshold of this frontier, it is clear that the dialogue is not merely a scientific curiosity, but a vital thread in the tapestry of being. The gut and brain, locked in ceaseless conversation, shape our appetites, our moods, our sense of self. The tools of the trade are our lanterns in the darkness, casting light on mysteries that have lingered in shadow since the dawn of sentience. As we peer further into the depths, listening, measuring, decoding, the next frontier beckons. For every answer unearthed, new questions emerge. For every technique perfected, a new realm of complexity unfolds. The journey is far from over. The dialogue continues, and tomorrow's discoveries wait in the silent spaces between heartbeats, in the restless stirrings of the gut, in the dreams that flicker behind our closed eyes. How quietly, in the hush of the night, the mind wanders through its own corridors, seeking answers to questions it scarcely dares to utter in daylight. The self, this peculiar tapestry of thoughts, memories, urges, and sensations, waits, poised on the edge of sleep, for some gentle revelation from within. Tonight, the journey has brought us to a threshold where science and philosophy entwine, the uneasy, wondrous intersection between the gut, the so-called second brain, and the elusive concept of human consciousness. It is here, in this dim-lit region, that the ancient and the modern, the material and the mysterious, whisper to each other in a language of nerve and wonder. For centuries, the heart was thought to be the seat of the soul. Later, the brain took that place, enthroned as the master controller, the origin of identity, the wellspring of thought. But science, in its slow, persistent unfolding, has revealed a more intricate landscape. The enteric nervous system, that dense web-like network laced through the gut, is not merely a passive recipient of the brain's commands. It is a vibrant, autonomous actor, pulsing with its own rhythms, shaping not only the body's digestion, but also its moods, its appetites, and perhaps, just perhaps, its sense of self. Consider for a moment the way you feel when anxiety knots your stomach, or joy sends butterflies fluttering through your belly. These are not mere metaphors. The gut's nerves, numbering in the hundreds of millions, are in direct conversation with the central nervous system, sending signals back and forth in ceaseless chemical dialogue. The vagus nerve, that great bidirectional highway, transmits not only instructions from the brain to the body, but also a flood of visceral information upward, shaping the mind's perception of the world. But is this conversation only a matter of physical sensation? Or does it extend into the realm of consciousness, into the ineffable territory of personhood and choice? Here the questions grow thorny, and the science, though robust, falls into the company of philosophers and poets. The second brain theory, as it has come to be called, suggests that the gut's nervous system is more than a relay for digestive orders. It is capable of reflexes without central oversight, able to sense, decide, and act independently. In the laboratory, when researchers severed all neural connections between the gut and the brain, the gut continued to orchestrate its symphony of contractions, secretions, and absorptions as if guided by an invisible hand. It learned even to adapt to new routines. These observations, though grounded in the material, carry a faint shimmer of the metaphysical. What does it mean, after all, to have a mind distributed through the body, to be, in some sense, a colony of intelligences rather than a single sovereign? Let us trace this thought further, as quietly as a dream's unfolding. The human connectome, the great intricate map of all the brain's neural connections, has become a central metaphor for understanding the self in the age of neuroscience. Each line, each junction, represents a whisper of information, a possibility of experience. The connectome is not fixed. It shifts with every memory, every learning, every brush with the world. It is, in essence, the living map of who we are. But what if this connectome is not only in the cranium? What if it extends in diffuse and branching lines through the core of the body, into the gut, the heart, even the skin? The idea is both unsettling and exhilarating. For if the mind is not locked in the skull, but spills outward into the viscera, then consciousness itself may be more than the sum of synapses. It may be a process, a weaving of multiple voices, some loud and articulate, others murmured in the secret language of neurotransmitters and hormones. Within this framework, the gut emerges not as a mere appendage, but as a participant in the creation of self. The enteric nervous system has its own memory, its own capacity for learning and adaptation. In animals, and tantalizingly in humans, experiments have shown that the gut can remember patterns of nutrient flow, adjusting its behavior based on prior experiences. These are not memories in the narrative sense. No one recalls their lunch through the gut's eyes, but they are nonetheless forms of stored information shaping future action. If the gut can learn, can it also feel? The question seems at first naive. Feeling, after all, is the province of the conscious brain wrapped in its layers of cortex. And yet, the gut responds to emotional states, and in turn influences them. The microbiota, the trillion-strong community of bacteria and other life forms that make their home in the digestive tract, produce neuroactive compounds, from serotonin to dopamine, which bathe the enteric nerves and, by extension, the brain. Changes in the gut can lead to changes in mood, and vice versa. This is no metaphor, but a documented axis of communication, a two-way street paved with biology. How far does this connection reach? Some philosophers, peering into the dim light where science and speculation meet, have suggested that consciousness is not an all-or-nothing property, but a gradient, a spectrum that emerges from the complex interplay of neural systems, both central and peripheral. The gut, in this view, is not conscious in itself, but forms a necessary substrate for the experience of consciousness. It is a co-author of the mind, providing the raw materials of sensation, emotion, and intuition. Here perhaps we find a new humility. The self is not a solitary monarch, issuing edicts from the high tower of the brain. It is a parliament, a chorus, a shifting alliance of voices, some ancient, some newly evolved, all speaking in the shared language of the body. The gut's voice may be quiet, but it is persistent, shaping our desires, our fears, our very sense of what is real. And what of identity? The old view that the self is a fixed and singular thing has given way to a more fluid understanding. The self is not a noun, but a verb, a process of becoming, stitched together from memory, sensation, and choice. The gut, with its ceaseless activity, its uncanny autonomy, is part of this process. It grounds the mind in the body, provides a context for thought, a place for emotion to land. Some traditions have long intuited this truth. In Eastern philosophies, the hara, the energy center in the belly, is the seat of will and intuition. In Western idiom, we trust our gut. We feel sick to our stomach with worry. We experience gut reactions that precede conscious thought. These phrases, born of lived experience, echo the findings of modern neuroscience. The gut is not a silent partner, but a co-creator of meaning, and yet the mystery deepens. If the self is distributed, if consciousness is an emergent property of many systems in concert, then what becomes of free will? Are we, as some skeptics suggest, merely passengers in a body governed by chemical impulses, our choices prefigured by the secret dialogue of brain and gut? Or is there, even in this complexity, a space for agency, an opening where the mind can steer, however gently, the course of the body? Some neuroscientists argue that the gut's influence is not deterministic, but modulatory. It shapes the probabilities, biases the system, but does not dictate the outcome. The feeling of hunger may make food more appealing, but it does not compel the hand to reach for bread. The flutter of anxiety may color perception, but it does not control the mind. In this view, the gut is a partner in the dance of decision, not a puppet master. Others, more radical, suggest that consciousness itself is a process of negotiation between competing signals, bodily, emotional, cognitive. The self, in this sense, is a temporary truce, a momentary alignment of interests. The gut, with its slow rhythmic pulses, provides a kind of background music, setting the tempo for the mind's improvisations. From this perspective, the search for the human connectome, the complete mapping of all our neural interconnections, becomes more than a technical challenge. It is a quest to understand the very nature of personhood, to discover how the many become one, how the disparate signals of body and brain are woven into the seamless fabric of experience. The connectome is not only a map of the brain, but a map of the self, a cartography of consciousness that must include the twists and turns of the gut. Some researchers dream of a day when this map will be complete, when every synapse, every axon, every neurotransmitter will be charted and the secret of consciousness laid bare. But others, more cautious, remind us that the map is not the territory. Consciousness may not be found in the wiring diagram, but in the dynamic interplay, the ceaseless dance, the emergent patterns that arise from complexity. The gut, in its quiet way, reminds us that understanding is not only a matter of analysis, but also of synthesis, of seeing the whole, not just the parts. The second brain is not a rival to the first, but a collaborator, a co-author of the story of the self. Its influence is subtle, pervasive, sometimes hidden, but always present, shaping the contours of thought, the textures of emotion, the very sense of being alive. As night deepens and the mind drifts towards sleep, the boundaries between body and mind, between self and world, begin to blur. The gut, with its pulsing rhythms and silent wisdom, becomes a kind of lullaby, soothing the anxious brain, grounding the wandering thought. The second brain whispers its secrets in the language of sensation, reminding us that consciousness is not a solitary phenomenon, but a symphony, a weaving together of many melodies, some bright and clear, others low and mysterious. And so, as the journey into the world of the gut draws on, the questions become not smaller, but larger. What does it mean to be a self, if the self is a process, a dialogue, a constellation of systems in conversation? Where does the mind end and the body begin, if every thought is tinged with sensation, every emotion rooted in flesh? Perhaps the search for the human connectome is not only a scientific endeavor, but a philosophical one, a quest to understand not only how we think, but what it means to be. The gut, in all its complexity, is both a mirror and a mystery, reflecting the tangled beauty of the self, hinting at truths that science has only begun to fathom. In the quiet darkness, as sleep approaches, the mind rests on these questions, not in search of final answers, but in gratitude for the wonder that remains. The gut, the second brain, pulses on, a gentle reminder that we are, each of us, more than we seem, a tapestry woven of nerve and memory, sensation and thought, body and mind, forever in the process of becoming. And somewhere, just beyond the reach of certainty, the next question waits, beckoning us onward, ever deeper, into the mystery of the self. As our journey through the whispers of the second brain gently drifts to a close, may you feel the quiet harmony between mind and gut, like a comet tail fading into the hush of space. Rest in this cosmic connection, and when you return, more Dreamtime tales will await to softly unravel the mysteries of our wondrous universe.